It's my pleasure to introduce Joel, the illustrious Joe. I don't think Joel, he doesn't need an introduction, but Joel, he was just telling me that he joined Moffitt in 2017. It's crazy. Yeah. That's crazy. So I can't believe it's been that long. That's awesome. But met the illustrious Bob in 2008 and Sandy in 2009. So a long history with lots of fruit from that collaboration. And welcome, Joel.
Hey, thank you so much. Can everybody hear me? Do I turn this thing on? Is this working? Okay, I'll try not to be too loud. Anyway, well, thank you so much. And Kristen, I believe we met in 2011 or 2012. Yes, at that time there. Well, thrilled to have the opportunity to be able to speak to you, the didactic learning, and then the morality. It's been so fun swimming in the Gulf of Mexico. I hope all of you have been enjoying it. So my background, just want to share a little bit. I am an evolutionary ecologist and I'm a game theorist. So mathematically, game theory makes me happy. Much of my career has been spent doing modeling, but also what we call mud and boots ecology. And my introduction to the field was thanks to Bob and then Tom Vincent, my game theory mentor. And it was at a conference, International Society of Dynamical Games. And indeed, that was where Bob and Tom finally agreed to let me work with them. They were willing to take the risk. And indeed, we outlined our first paper on a napkin. We outlined our first paper on a napkin, and it's been really gratifying to see how that topic, how the double bind therapy is sort of gaining traction. And then it was Sandy. We had as my boss at Moffitt right now, is our moment was, will not surprise you, those that know me, was late at night. Midnight, in fact, Sandy and I closing down the conference in this town square, drinking and chatting and sharing kind of our vision of things. And then, of course, these kind of interactions continue to this day. Exactly the sort of networking that I've enjoyed thoroughly with all of you, and looking forward to another evening. Just want to share my lab with you quickly. So these are the folks that are currently in my lab. All of them, I believe, are here except for Kieran, and then Chris Whalen might be on. He may be on Zoom. He lives in Chicago. Many of them had posters, and hopefully you've had a chance to meet them. This is sort of the bill of fare, what I'm going to try to do today. Why cancer, ecology, and evolution? A bit of Darwin, principles of ecology and evolution that may be useful, ecology of fear, and then mechanisms of coexistence. So first off, why cancer, ecology, and evolution? I mean, it really comes down to the definition. So for me, I've always found it fascinating when you ask people, what is the definition of cancer? Even the cancer biologists will waffle, or they'll give you some sort of answer. And so working with this wonderful team of people, we went about updating the definition of cancer. And I'll get back to why I think that was an important thing to do. We recently published a paper, and here is our proposed updating of the definition of cancer. A disease of uncontrolled proliferation by transformed cells. And then the kicker, subject to evolution by natural selection. Soon as it's subject to evolution by natural selection, that completely alters the way you think about it, approach it, and I would venture that all of the talks I have heard in here, whether you like to have a definition pinned on you, you may not, and I completely understand if you don't, but essentially every talk is operated as if this is the definition of cancer. Because here are the, what's interesting is even going back to Hamburg, they even identified the fact that the hallmarks of cancer are analogous to Darwinian evolution. We just published this year this paper where we dissect the hallmarks of cancer and place them solidly in an ecological and evolutionary framework. Rangini, my grad student, thanks very much. And now so to me then, more specifically then, cancer is a speciation event. It's where a cell that was once part of the whole organism jumps levels of selection, and at that point, its tumor becomes the ecosystem. It evolves according to the laws of ecology and evolution. And the difference is, because it's not an infectious disease, except in very rare cases, the tape of life gets replayed. And so you can really then examine cancer exactly the same way we do ecosystems. They're hierarchical, from molecules to the whole biosphere. They also then have complex dynamical system. And then the key bottom one down there is They're the product of natural selection. So this kind of is the parallels as to how I kind of see cancer. So that means cancer is the most personal experience that any of us or our friends and family members wherever will ever of natural selection. So I'm going to define a few terms. Evolution. Change in the heritable characteristics of a population. Change in the heritable characteristics of a population. Ecology, the study of the interaction of organisms with their environment. And me as an evolutionary ecologist, evolutionary ecology is studying life through its adaptations as a product of natural selection. Now, this one is how I like to view natural selection. Natural selection is the force of evolution that promotes adaptations. The other forces of evolution, neutral drift, mutation, gene flow, but natural selection is the only force of evolution that actually promotes adaptations. What are adaptations? They're traits, heritable traits that maximize fitness given the circumstances. So very contingent, depending upon the environmental circumstances. And then finally, what is fitness? The definition down the road, fitness is per capita growth rate. If you're dealing with complex life histories, like some of those intestinal parasites that go through different species and do crazy things, defining fitness can be tough. But for cancer, it's easy. It's just simply proliferation and survivorship basically defines per capita growth rate. Darwinian evolution has three elements. Heritable variation. Like begets like, but with boo-boos. So my daughters look just enough like me that I know it wasn't the postman. But thank God they don't look completely like me. They're beautiful like their mother and look much better. Heritable variation. The second is struggle for existence. That's a statement of ecology. We will get to that. Struggle for existence. That's the ecological stage on which plays the evolution then plays out. And finally, this heritable variation has to influence fitness. My favorite for showing that is this slide. Do we have dog lovers in here? Cool. Do we have people in here that prefer cats? Okay, so I'm going to go to the cat person here. Would you agree that sometimes when you see some of these little dogs on strings, it's like almost an affront to evolution? That this thing can even exist? Teacup chihuahuas? Now you guys are going to hate me, but we've been here, we've become friends. One of my recurrent fantasies, I will not act on it. It's okay to think about things, is doing and take a small dog and have it on a leash swimming behind and see what the alligators do. No, I'm sorry. But the key point of this slide is the wolves. Would they even imagine that all of this was part of their heritable variation? All of it was part of their heritable variation. Wolves think, well, actually, I know what they would think of a small dog. What happens? You have all that heritable variation, and then it gets pit against ecological reality. And against ecological reality, this experiment has been done. There are wild dogs, the dingo being the most famous in Australia, but Smoky Mountains, the Middle East, Southern Africa, and they all start like that, and they all sort of become dingo-like. They all converge on a similar strategy. So what that means then is, in terms of heritable variation, let's think about cancer. There's heritable variation in spades. Not necessarily the traditional beanbag genetics, where you think of a mutation, big T allele, little T allele, can I roll my tongue? Some of you are going, yep, I still have it. No, I still can't roll my tongue, darn. But in fact, heritable variation for cancer cells can be epigenetic, copy number variation, Noemi. It can be whole genome doubling. It can be backing off in terms of aneuploidy. It can be parasexual, like Andre's work that he's done with cell fusion. So cancer cells have the whole human genome at their disposal, and they can explore that genome epigenetically, upregulate, downregulate. So what that means then is by the time you pit that heritable variation of a cancer cell against their ecology, really like nature, really, cancer and nature are a rather dull subset. They're exciting, but a rather dull subset of what is evolutionarily feasible and what is ecologically acceptable. That brings us then to cancer following the laws of ecology and evolution. First off, and I'll be thinking about solid tumors. You can make extensions to the liquid tumors, but basically a tumor is sort of a viscous chemostat. You've got a clump of cells, all manner of things that form sort of an ecosystem. Blood flows in, does its thing, and a certain amount of those liquids flow out. What happens inside that tumor is threefold. There are behavioral dynamics. The cancer cells can acclimate. They can show phenotypic plasticity. Interestingly, as a trait, phenotypic plasticity is actually a compound trait. It means the ability to assess and then the ability to respond. So anytime you're talking about phenotypic plasticity and trying to wrap your arms around it as an adaptation, remember you're really talking about two aspects. What can the cancer cell assess and how is it able to respond? Those are the behavioral dynamics. Then there's population dynamics, births and deaths, and finally evolutionary dynamics. And the first law of ecology that applies, we've seen this today, or we've seen this over the couple of days, exponential growth. All populations have the capacity to grow exponentially under ideal conditions. Roughly speaking, cancer cell lines can grow or double about every 24, 28 hours. But do they? So the Fisher-Skellam equation is very nice. It was developed to study the range expansion of species. And the Fisher-Skellam equation has R. So if you know the intrinsic growth rate of the species, you know something about its dispersal, you can actually predict how far and how fast it will grow. Rabbits in Australia, they were spreading at an enormous rate, 50 kilometers a year. 50 kilometers a year. The average rabbit doesn't even move that far. They were moving at their intrinsic growth rate. That says the habitat beyond was perfect, ideal, and they did. Another species near and dear to my heart, for all those that come from England, the gray squirrel, is our revenge for the European starling. And the gray squirrel spread also quite quickly. It spread about 5 kilometers per year, and that's about 1/3 its potential, 1/3. So that means the English countryside wasn't perfectly ideal. Why? Because of competition from the European red squirrel. And then there's my favorite in thinking about this, and that's prairie dogs. Prairie dogs can breed just as much as rabbits. They can breed as much as squirrels. They are a ground squirrel. But look at this, 30 meters per year. Another study showing range expansion of prairie dogs, a whopping 40 meters a year. So we've seen 50 kilometers, we've seen 5 kilometers. Why are they so slow? And the answer actually is this wonderful paper, and it deals with what are the factors that influence the ability then of a population to expand. And I think these were all meant for ecology, but I think if you take a gander around all of these things, beyond the tumor edge, all of these factors, thank goodness, slow it down. It seems to grow fast, maybe several percent per day, sometimes 0.5% per day. We've all seen the growth dynamics of tumors, but that growth dynamic is way smaller than their potential. In fact, then, really, tumors are not like rabbits. They're not like squirrels. They're actually more like prairie dogs. You have often a very sharp boundary. I had to sit around for hours to get this photo because I wanted prairie dogs to be out of their holes, and I wanted to be able to show you just how tight the boundary is. Isn't that astonishing? You can draw a line between where the prairie dogs are and where the prairie dogs aren't, in many ways like you can with tumors. And probably, I don't have time to discuss it because I want to get through a lot of slides, is probably the same things that prevent the prairie dogs from spreading. Look at that one prairie dog that could just hop its way over there if it wanted to, but it won't, is the same thing for cancer. And so with tumors, it's interesting then what's beyond the tumor that's slowing it down or speeding it up. Struggle for existence. And that is no population can grow exponentially forever. There are limits to growth. And that brings us to that gnarly term K. How many times have we heard K? What the hell is K? Well, it's a breakfast cereal that actually there's 3 Ks. And I just want to walk you through kind of a thought experiment to be able to sort of see. So it deals with density dependence. There's density dependent feedbacks, there's limits to growth. And I'm going to pause it. And I know Sandy has published papers very early on picking up this issue. There's really sort of three Ks. The first K can come from any of these models. Look, they all have K. Pick your favorite model of population growth. We've seen them. But then the first K is that within a tumor, the cancer cells then can reach a certain density. That density may fluctuate depending upon what's going on, the necrotic zone and whatnot. That probably happens very quickly. Achieving sort of that local carrying capacity and fluctuations in that, that may happen in a matter of days, a week. The next one is, what constrains the growth of the tumor itself? It needs blood supply. So here's a really simple model, actually drawn from Yoda's law, which is called the 3-2 thinning law. And that is, you're imagining resources coming to the tumor. You acquire new blood vessels, and notice I made it to the 2/3. And that is, as a tumor grows, it doesn't acquire blood proportionate to the population size of the cancer cells. So I'm going to use 2/3 scaling, and then the cancer cells are sucking up the resources, and then there's some efflux. Now we make a simple consumer resource model for the cancer cells. They harvest resources, they turn them into babies, and they have a death term. And if you make that model, you get a new carrying capacity. You can actually calculate how big the tumor can get. So that means the tumor has a maximum size. Now you get the maximum size that the tumor can be. But there's metastasis. And with metastasis, there's the ultimate carrying capacity. And that is, you can think of metastases are basically cancer cells colonizing new sources of blood. They're acquiring access to new sources of blood. That's why, thank goodness, there aren't very many lipomas. There's really no blood there. Yay. So what happens? The human body, we turn over cells at about 0.1% per day. You die if you have to turn over your cells or feed cells to turn them over at 0.2% per day. So the ultimate carrying capacity, if the cancer doesn't cause organ failure like glioblastoma or the failure of the liver, but if you're just feeding it, there is an upper limit, kind of like the Little Shop of Horrors, if any of you remember Audrey 2. So what are the limiting factors? And I think this is fascinating. You can talk to people, obviously we know oxygen, pick your favorite nutrient, glutamine, cysteine, glucose, what are they? Obviously the food they get is whatever comes through the blood. Furthermore, I think a really exciting area that we study in ecology is nutrient cycling. So the glucose probably goes around once. It goes in, gets burned, CO2 comes out, it's gone. Glutamine, it may go around a few times. Some of the glutamine might get burned, some of it gets used to build stuff, and whatnot, it gets recycled. Cysteine likely gets recycled a lot. And so this notion of nutrient cycling, we kind of picked this up with sort of cancer foraging ecology with Sarah, and that is, you've got goodies come in from the blood. You also have cell-cell competition. Here you have co-feeding with the fibroblast or other cells, and then you've got macropinocytosis, where cancer cells then basically forage on their dead neighbors. What can you learn about foraging? So we're going to let this little cute guy, the elephant shrew, teach us something about foraging and see if we can learn something that we might want to know in cancers. So elephant shrews are wonderful. They got long tongues and long noses. They poke around in rocks, and they stick their tongue through the rocks. Can you imagine sticking your tongue into holes? And then what they're looking for is insects, and then they pull out the insects. So we were interested in what foods do they eat, resources, and what substrates do they like. So we set up sand, we set up sawdust, we set up rocks, we tried sunflower seeds, mealworms, and alfalfa pellets. So imagine what they might like best, what would happen. And then here is just the data. And I hope you can see how this actually applies to thinking about cancer. This is the proportion of food harvested by type from each of these patches. Unsurprisingly, they definitely like pebbles better. They like sticking their noses in rocks. They did not like sand or sawdust. Interestingly, we thought they would like mealworms best. Nope, they love sunflower seeds. Then mealworms, and they really are not vegetarian. They're not keen at all. And so what it means then is here you've got different foods, different substrates, and what does it mean? We should expect cancer cells, their nutrient uptake should be strongly based on resource supply, what you put out in the patch, the type of nutrient, and the extracellular matrix. And I would venture that that exciting area of research is only just beginning. Then there's the immune interaction. Who can't love it? Predation. So interestingly, the immune system, thanks to Kim Luddy and many others, oh my gosh, how many would you agree it's complicated? Now that is a messed up predator-prey system. So basically, to me, the immune system is a policing bureaucracy. It's trying to balance, kill the bad guys without killing the good guys. Trying to prevent the kind of autoimmunity we're seeing in Chicago now with the ICE agents picking up everybody. And then you've got the cytotoxic immune cells aren't quite predators because they don't work on commission. A standard predator kills its prey, gets the goodies, and that allows it to make more babies. But cytotoxic T cells don't work on commission, so that makes them a bit odd. But cancer cells do exhibit fear responses. So what does that mean? So if you'd like, in this paper, we explored the three principal sorts of physical defenses that organisms have. You can have nasty chemicals, you can have spines, and you can have armor. Each of these is the predator in different ways. Each of them has a different effect on the predators. And if you go across mammals, fish, insects, microorganisms, and even cancer, you can find examples of all three of these types of defenses. That brings us to the ecology of fear. And the ecology of fear is really what my career was built upon. And that is the non-lethal effects of predators, how the mere presence of predators, the fact that the predators could be there, how that influences the evolution, the behaviors of the organisms. And I want to give you an example of it. You can actually calculate how frightened a cancer cell should be. You can calculate how frightened you should. And that is, how frightened should you be? This is called the activity cost of predation. It has three components. So the next time you want to come visit me in Chicago and you're saying, how frightened should I be? Type in, what is the risk of something bad happening? The second is, what do I have to lose if something bad happens? And then you divide it by how fun is the activity? How valuable is the activity? And so if somebody comes up and says, I am too frightened to do that, are they telling me they see it as risky? Are they telling me they have nothing to live for? In which case we do have mental services, we do have counselors that can help you reevaluate your sense of value, or you just don't think the activity is very fun. And organisms respond tightly to that. And I'm going to give you an example of how it works and what it might mean. So I'm going to take you to South Africa. You have these little patches of trees. We're going to be focusing on these patches of trees that are called islands of fertility. They have a bit more fertile soil, so they support this vegetation. We're going to have Mohammed Abu Baker. He's currently a faculty member at the University of Amman, Jordan, who did this work. As you can see, he is not afraid of snakes. That's a puff adder that came into camp. And then we're going to work with these guys. What do you think? On the cuteness scale, this is the striped mouse. And we could set up food patches. The striped mice are actually very social. They like to forage together. You can see it in this particular picture. And what we did is we were asking the question, what do they think of the structure of their tumor microenvironment? Now, when you trap them, we never, ever trapped any inside here. Never, never. And almost all the trap was out here 6 to 12 meters away. So there could be two reasons. One is there's no food. There's no point. Or maybe it's deadly. Maybe it's deadly. So we did the same kind of approach that I talked to you before, and we measured, we'd put 35 seeds in a dish, and we'd come back and see how many of the seeds are left. And we put them inside, and we put them all the way to 5 meters. And what you see conspicuously is they will not feed. Even if you offer them yummy food, they will not feed. They have self-control. They won't go into those islands of fertility, are really islands of despair for the mice. And then you can see they're really quite happy feeding. They'll take over half the food out of those patches once you move around. So we could use this to actually make a map. You can actually see where the tumor is hot. Every place that's red is where they are too frightened to go. Any place that's green, that's a cold tumor. For whatever reason, they feel safe. That yellow zone is sort of in between. We could map it spatially. But then the question is why? And I think this is what's exciting about the ecology of fear, is you get these direct and indirect effects. So we thought it was odd. Generally, mice and rodents like cover. They're supposed to like to be hidden among the trees. Well, it turns out this animal, the genet, you can see sniffing one of the patches. This animal, the genet, loves striped mice. The genets, though, for curious reasons, are always in the islands of fertility. They barely come out. They barely come out. And we were curious, well, why is that? It turns out they're afraid of these, the leopards. And so these are the exciting kinds of interactions that you can think of in the immune system beyond just these sort of straight dynamical models. There's a whole ecology of fear going on. And in this case, to understand the mouse, to understand why the mouse felt about its habitat in this way, you need to know the predator, the genet. You need to know what the genet is afraid of, in this case, the leopard. Next, cancers are heterogeneous. And I love that because that gets into niche filling. And much of my career has been a community ecologist. Community ecology comes down to these scales of diversity. And we see these same scales of diversity that we measure in ecosystems. We see them in cancer. People measure them. It's exciting. You can probably recognize your scale up there. And furthermore, coexistence comes down to the jack of all trades is a master of none. Trade-offs. If there were no trade-offs, you would get what we call a Darwinian demon. You would simply get the cancer cell that is the best at being a cancer cell that outcompetes all other cancer cells. And so we've seen examples of trade-offs and talks. These are the categories and mechanisms of coexistence. Isn't that cool? Just about every coexistence of organisms can be binned into one of these 5 categories. Sometimes it's a mixture of them. We've seen them in the talks here. It's been so fun. Diet choice, eating different things, habitat selection, living in different places. Food safety trade-offs is rampant in nature as well as in cancer. We saw variance partitioning is my favorite. Why? That was part of what my PhD dissertation was about. Some of the first models of how variability, we've talked about, how variability can promote coexistence. And then finally, competition colonization trade-offs. Does anybody recognize that from talks yesterday? How can we see these mechanisms? Well, I told you I study them. This is what I do. So here are my friends. These are all the squirrels you can find around Chicago. Isn't that cool? It's a biodiversity hotspot. And you can find all of these. So the woodchucks, they tend to eat grass and green vegetation. The 13-line ground squirrels like to eat seeds. Habitat selection. 3 of these guys are ground squirrels. Well, you can guess where they live. 3 of these guys are tree squirrels. Crumb pickers versus cream skimmers. The gray squirrels in Chicago, they stick it out all winter. Doesn't matter what the weather is, how cold it is, they stick it out. They're what we call crumb pickers. The chipmunks, they're rude. As soon as things get bad, they go to sleep. They sleep through the bad part. How many of you wish you could sleep through the bad parts of life? Lessons from a chipmunk. So they sleep their way and then they arrive in the spring when things are good again. If I'm a gray squirrel, that's got to really piss you off because they're cream skimmers. They show up, take the good stuff, and go to sleep, and you're left whatever they leave. And then my favorite, come by my office, if you're going to be there at the IMO workshop, is the fox squirrel and the gray squirrel. I have stuffed taxidermy-mounted examples of these, probably the only taxidermy-mounted mammals in any cancer center around the world. These 2 are great. The gray squirrel is highly competitive, but very prone to predation. The fox squirrel is not a very good competitor, but it's really good at avoiding predators. In fact, the fox squirrel is orange to tell the predator, I'm not a gray squirrel. How's that for cool? So the coloration of the fox squirrel can only be understood in relationship to that. And if you're a fox squirrel, you want the predators to know I'm badass. It's a gang sign. Does it happen in tumors? Yes, we've seen some examples. Mark Lloyd examined this on the edge in the interior of breast cancer, used 2 biomarkers, CA9, CA12. I've never gotten data this good with animals. On the edge of all of these 15 women, very early, CA9 was upregulated heavily on the edge, and then in the interior, CA12, and nobody upregulated both, virtually nobody both. It can also be microhabitat. So I'm going to share with you my academic granddad, Robert MacArthur, and he showed that you could get the coexistence of these very similar warblers by partitioning parts of the tree. How's that for fine-grained habitat selection? That there's trade-offs. Some of them, Chris Whalen studied this, are better at getting insects from the bottoms of leaves. I hope you're listening, Chris. Others are better at getting them on the top of leaves. How's that for weird? Some. And that kind of partitioning we also see. So in the case of Bob, one of our favorite people, Khalid, we worked on this idea of seeing that where you've got blood vessels, you have a higher density of cells. And notice, as you move away from the blood vessel, you have a completely different class of cancer cells. You get a sharp break in the cancer cells, just like you get, this is an aerial shot of my PhD study site in Arizona, where you have the wash and the vegetation. Then as a final bit, there's feedbacks. The feedback environment is so important in tumors, which is why tumors don't look anything like normal tissue. Remember that prairie dog town? Did it look like normal vegetation? No. So think of the prairie dog, and that comes down to this topic area that I think is so important and interesting, and that is 3 things that happen in tumors that happen in nature. And I just want to offer you some terminology that may be helpful if you want to probe the ecological literature. There's what is called ecosystem services. That is where organisms, not intentionally, just by the way they modify their environment, help out other species. So trees growing on hillsides hold the soil. And by holding the soil, that allows other plants to live there, allows other animals to live there, which is why if you cut all the trees off of a hillside, you get mudslides. Similarly, I want to make a distinction between ecosystem engineer and niche construction. I've heard both of them in the last couple of days. Ecosystem engineer is simply a species that in its normal activities has an unusually large impact on the environment. It may be accidental, like a bug on your windscreen, or it may be adaptive. And so it is just like an elephant tearing down trees when they're hungry to get the leaves. That creates a wooded savanna. The elephants don't tear down trees to make a wooded savanna. They're hungry, but it changes the whole ecosystem. A subset, now this is key, a subset of ecosystem engineering is niche construction. And that's where an organism, as an adaptation, modifies its environment in a way that makes it more suitable for itself. So the beaver, of course, is the famous example then of niche construction. I could go on and on. We heard about evolutionary rescue. I love it. I didn't even have a chance to talk about game theory, but I hope you've kind of got a sense that there are some really amazing parallels between ecology and evolution over here and ecology and evolution over there. Thank you very much.
We have time for maybe one quick question. Yeah.
Yeah, thank you very much. It was a really great talk. I enjoyed it a lot. So my question is a little bit, how far can you take this analogy? Because obviously in real ecosystems, the predators evolve and the prey evolve, so you have co-evolution and it balances a little bit. But then in the cancer, obviously the cancer cells are evolving, but the healthy cells are probably not, or at least they should not, otherwise they would be cancer, right? So how does this change the analogy and what implications are there for that?
I don't think it alters the analogy at all. What it does is just means cancer is more of a subset of everything we happen over here. So everything happening over on the right side, the processes and whatnot may not be going on in the cancer. And so what happens in the cancer then is right. The normal cells, they're props. They're very sophisticated props. They're very dynamic props. They do stuff. But yes, they are not players in a typical sense. The immune system is interesting because that's the one place where our body uses evolution to fight evolution. So that case, then there are some interesting parallels. So I think what it is you describe what you see as the ecology of your system. And I promise you I can find something on the right side that matches it. So no, I think the analogy works great. It's just that the left side, by its very nature, is going to be a subset of what we see on the right side. Did that answer your question? Was that?
Yeah, thank you.
Done.
Well, let's thank our speaker.
Okay, very good.